The timer of tamoxifen, otherwise known by brand names such as Nolvadex or its original compound ID ICI 46474, is one that stretches back to the 1950s. It is a derivative of triphenylethylene that demonstrated hormone-altering effects, specifically on estrogen. This led to a new pathway of drug discovery that would revolutionize the treatment of breast cancer. With the combination of cyclophosphamide, methotrexate, and fluorouracil chemotherapy, tamoxifen initiated the greatest decline in case fatality throughout the 1980s. But what makes tamoxifen special? First, it is considered a first-in-class drug, the first selective estrogen receptor modulator, or SERM, and it enabled the specific targeting of ER-positive cancers. Second, is that due to the difficulty of treating this cancer, the revolution in treatment and significant impact on mortality resulted in ICI Limited Pharmaceuticals Division being presented with the Queen's Award for Technological Achievement by the Lord Lieutenant of Cheshire on July 6, 1978. 3. A unique fact about tamoxifen is that, as you will see as we walk through the timeline, tamoxifen was initially targeted as a contraceptive, not an anti-cancer drug, and the intention almost led to ICI Pharmaceuticals scrapping the development altogether. Once ICI 46474 was not found to have ovulation-inhibiting properties, it was only through the protestive resignation of its head scientist Arthur Walpole the development was picked up again, and thank God it was. In the late 1950s, there was a surge in the research and development of a novel class of anti-oestrogen compounds as a potential for a morning-after contraceptive pill. At the time, Imperial Chemical Industries, ICI Pharmaceuticals, and Arthur Walpole, a reproductive endocrinologist, led a team in hopes of developing this new class of contraceptive. In 1962, a chemist in Walpole's research team named Dora Richardson was developing derivatives based on a triphenylethylene molecule and synthesized the world's first compound with the name ICI 4647, which would then later become tamoxifen. Originally, tamoxifen was intended as an estrogen inhibitor. However, during later drug testing trials, it was found that tamoxifen had no efficacy as an estrogen inhibitor. On the contrary, it stimulated ovulation in women. The further development of tamoxifen was abandoned by ICI Pharmaceuticals as a futile effort, as previously mentioned. However, research team leader Arthur Walpole insisted that ICI 46474 could be a potential therapeutic for breast cancer. Subsequently, ICI 46474 was trialed as a breast cancer therapeutic and proved to have efficacy in stopping cancer cell growth and proliferation. Unlike conventional drug discovery processes, the target for tamoxifen was not selected first. In contrast, it was discovered. It was found that the principal mechanism of action of tamoxifen is mediated by its binding to the estrogen receptor and the blocking of the proliferative actions of estrogen. However, it is also suggested the anti-proliferative action of tamoxifen is the induction of the synthesis of the cytokine-transforming growth factor B by tamoxifen, which acts as a negative autocrine regulatory molecule. Following the synthesis of triphenylethylene derivatives by Dora Richardson, the compounds including ICI 46474 or tamoxifen underwent lead identification. Tamoxifen has a geometric cis isomer, compound ICI 47699. Following analysis, it was identified that these two isomers had opposing actions, with tamoxifen demonstrating anti-oestrogenic activity and compound ICI 47699 having anoestrogenic activity. Considering the nature of the project, tamoxifen emerged as the lead compound and moved forwards. Tamoxifen was then developed into a citrate form for better pharmacokinetic profile. However, differing to conventional drug design, no optimization or alterations were done to the original structure. The drug then moved into preclinical trials. ICI Pharmaceuticals performed a number of preclinical pterogen studies using a range of species, and whilst rat infants initially demonstrated kinky ribs, these symptoms eventually resolved following birth. All other species studied found that tamoxifen was safe. and that the seemingly teratogen effect seen in rats was due to restriction of the uterine growth, and therefore making tamoxifen not a true teratogen. Studies on toxicology under GLP conditions were conducted using a number of species, including rats, mice, rabbits, dogs, monkeys, and sheep, and found to be non-toxic across all species. These studies were also performed for more than 6 months. Organ damage studies were also conducted using pigtail monkeys, where changes in the reproductive cycle most closely resemble humans. The compound was found to be non-harmful. The mechanism of action of tamoxifen was not known when clinical trials were initially conducted. Instead, following the Christie Hospital clinical trials, studies attempted to evaluate ICI 46474's mechanism of action, resulting in strides in 1973 by Worcester Foundation in animal tumor models, particularly the DMBA-induced rat mammary carcinoma model. These studies fully describe the anti-tumor activity of ICI 46474 in vivo. Reproductive studies were also performed on this compound, with compound ICI 46474 being found to be most effective in terminating early pregnancy in rats on the 4th day after mating. It therefore supported the hypothesis proposed by Walpole and Harper, in that compound ICI 46474 prevents implantation in rats by counteracting the oestrogen surge on day 4. However, despite the success in rat models, the relevance to humans who do not experience this oestrogen surge was questioned. Rather, anti-oestrogens, like tamoxifen, in fact improve fertility by inducing ovulation in subfertile humans, so much of the drug development was not relevant. The first collaborative clinical trials took place from 1967 to 1971. ICI began planning a trial with compound tamoxifen for the induction of ovulation in amenorrheic women, rather than as a contraceptive, as the results had been shown to not be relevant for humans. However, as the mechanism of action was at this time unknown, it caused fears for fetal malformation despite its lack of teratogenic effects in animal experiments. The lack of teratogenicity was considered as not enough of a safeguard to constitute moving it to human trials. Because of this, they believed that the first woman to undergo the trial should be offered an abortion in the case that there was a teratogenic effect. And to complicate this even more, the existence of the 1967 Abortion Act would severely limit the number of women that could be selected. So whilst they tried to plan around these complications, they still need to do therapeutic study to find the safe dosage range for use in contraceptive trials, and the approval for this was obtained from the CSM in 1969. The compound was approved for treatment of anovulation or menorrhagia associated with high levels of endogenous estrogen, and for the treatment of breast cancer in 30 menopausal and postmenopausal women. For the clinical trial regarding the treatment of anovulation or menorrhagia, tamoxifen was found to be capable of inducing ovulation rather than suppressing ovulation in women, despite the positive results in preclinical rat studies. But there were better and more exciting results from the breast cancer trials at the Christie Hospital, despite the goal of testing IC 46474 as a contraceptive. They found that the treatment of ICI 46474 was just as effective as traditional estrogen and androgen hormone treatments, and also had no toxicity and was well tolerated, even at the highest oral dose of 10 milligrams. The team was very excited by the results of the first trial, but their company ICI was not as enthusiastic. They told the team they were supposed to be looking for a contraceptive, not an anti-cancer agent. The development of the department also felt that it was hopeless. However, the CSM granted the company permission to prolong and extend the trials on the basis of positive clinical results. By the end of 1970, 60 patients had been admitted to Christie breast cancer trial, and of the 40 women who had been on the trial for more than 10 weeks, all had shown measurable and marked tumor regression. The clinicians carrying out the trial, Drs. Todd and Cole, also reported how impressed they were with the absence of toxicity and the low incidence of any side effects. So the previous clinical trial testing of ICI 46474 as a cancer treatment at the Christie Hospital was really decisive and made a huge impact. It confirmed that anti-estrogens could be used to treat breast cancer, and this catalyzed further studies to test anti-androgens in animal tumor models. These studies were first started in 1973 at the Worcester Foundation for Experimental Biology, using the DMBA-induced rat mammary carcinoma model. This model was considered state-of-the-art because no other hormone-dependent models were available for study. There was also another study at the Tenovus Institute for Cancer Research in Cardiff, which also used the DMBA-induced rat mammary carcinoma model for the study of anti-tumor actions of ICI 46474. These studies fully described the anti-tumor activity of the anti-estrogen in vivo, which was also very convenient as the efficacy of tamoxifen was being established widely in breast cancer clinical trials as well. The study of cancer as a broad treatment was demonstrated throughout the 1970s, with tamoxifen showing clinical applicability in a multitude of studies and consistently demonstrating reduced incidence with low toxic side effects. It also became prevalent as a preventative method in healthy women for those at risk of breast cancer, as it notably decreased incidence of breast cancer. The final point in our drug discovery timeline is registration and post-marketing surveillance. The patent for ICI 46474 was applied for in 1962 and approved in 1965. However, its patent protection was repeatedly denied in the US until the 1980s due to unknown reasons. Since then, there are patents involving compounds that are derivatives of tamoxifen, new processes for preparation of tamoxifen, and the use of tamoxifen in the treatment for specific diseases. Drug registration. Currently, there are two types of tamoxifen drugs in the market. Nolvadex, which is an oral tablet form of tamoxifen, which was approved by the TGA in 1977, and Soltamox, an oral solution of tamoxifen, which was approved by the TGA in 2005. In Australia, tamoxifen has quite a late entry into the market. Nolvadex only obtained TGA approval recently in 2016 and was entered into the Australian Registry of Therapeutic Goods not long after. Post-marketing surveillance. Since its approval by the Committee of the Safety Medicines in the UK in 1973 and by the Food and Drug Administrations in the US in 1977 for the treatment of breast cancer, tamoxifen has shown good safety and efficacy. It is now available in more than 110 countries around the world as a first-line endocrine therapy for the treatment of breast cancer.